Hello, today we are going to discuss about the pH determination by calorimetry. So pH is the negative logarithm of hydrogen ion concentration. So in which the concentration will be changing the physiological functions within the body. So how we can able to determine what is the pH present in the particular physiological function in the body, and this can be determined by using a calorimeter. In this today's topic, we are going to learn about the various methods to determine the pH, and especially the pH determination by using the calorimetric method. So first of all, we will need to understand why to do the determination of pH. That is, why the pH is measured. pH is measured because to test a sample against a legal requirement. So according to the industries, every each and formulation they are having a legal requirement in order to sell or use by any of the countries, by any consumers. So to test a prepared sample against any kind of the legal requirement, pH is very important play. Then to test a chemical against a specification. So to test the chemical agents and their uses against the specification which is given by the particular industries or by the legal requirements. Then this pH can be used as a part of analytical method, so which is used in the analytical method development. So analytically how the pH is going to be used, so it can be used as a analytical method tool. Then which is used in the process control in chemical industry to check how the process is going to be in a continuous manner, and how can we control the process of converting of one molecule to another, or in the internal process, or when we are making the intermediate products. So how can to control the process of manufacturing of chemicals in the chemical manufacturing industries. Then pH plays a very important role, and thereby the environmental monitoring of waste and effluents will also come into an existence and plays an important role. So it is very important to monitoring and controlling the biochemical reactions in the environment, and many of which only takes place in a particular and also sometimes in a narrow pH range. So it is also a convenient measure to find out the acidity or the alkalinity of a aqueous solution at a specific temperature. So at what particular temperature, whether the solution is in acidic condition or it may be in an alkaline condition, which can be measured by determining the pH of the solution. So this is used to measure on a continuous scale from 0 to 14. So 0 to 7, which considered as acidic pH, 7 as neutral, and 7 to 14 as a basic pH. So in a chemical laboratory, pH is commonly measured using a electronic pH meter with a scale of 0.01 pH meters. Now we will discuss with the various methods to determine the pH. So basically there are two important methods to determine pH. One is calorimetric method and the second one is the electrometric method, or which is also called as potentiometric method. So these are the pH can be measured by using number of devices or devices initiating systems, like you can use a pH meter, can use pH indicator papers and the litmus paper to know the pH of that particular formulation or the solutions. The first method we are going to discuss is about the colorimetric method. So colorimetric method, which will depending on basically on the color indicators and can also be determined by using the pH papers. So this colorimetric method basically involves to test the samples, the color intensity of the test sample with the standard color intensity and to find out the exact pH depending upon the intensity of the color produced in the solutions. So colorimetric analysis uses the variations as a means of determining the pH, since it requires to know the intensity of the color of a solution which keeps on changes with the change in concentration or change in the pH. So the color may be changes may be due to the inherent property of a substance in the solution. Inherent properties like color or the physical properties like solubility and temperature, then dielectric constants. So these are all called as the inherent property of a substance, or it could be a formation of the product as a result of addition of suitable reagents or acid-base indicators due to the addition of another reactant which can be turned into a change in the pH. So therefore, the pH of a solution can be determined by comparing the color intensity of the solution with unknown pH, and an indicator can be used to measure the pH of a substance. So the common indicators which are basically used in the determining the colorimetric, like using the Congo red, then phenolphthalein indicator, methyl orange, phenol red indicators, bromothiamine blue, bromocresol green, and also bromocresol purple indicators, which are commonly used pH or color indicators. So these are some of the examples for some acid-base indicators. So most commonly used methyl violet indicators. So which is when it is in acidic media, it gives yellow color. When it is in basic media, which gives violet color, that is the pH color change interval will be from 0 to 2 pH. Then thymol blue with pinkish red in acidic, yellow in basic, having a pH range from 1.2 to 2.8. Then Congo red in blue color, which is 3 to 5.2 pH, and red in basic pH. Then we have phenolphthalein indicator, which is commonly used, which having the pH range from 8 to 9.8, and it will be colorless in acidic region, but which will get changes into pink in basic range. So these are how these indicators will work. Okay, from pH 1 to 14, like methyl orange, from red to yellow, methyl red, from red to yellow again, with a change in the slightly pH range, from bromothiamine blue, from yellow to blue, that is from acidic to basic, the neutral red, from red to yellow, and phenolphthalein from colorless to pink color when it is turns into from acidic to basic. So in this colorimetric method can also be found out by using the pH papers. So where the pH papers were dipped into the solution and then finding out what is the color intensity which is produced on the pH papers, and those papers samples will be tested with the standard colors. So in which the pH 0 is red, 1 is red, and slowly it will turn into pH 7 with neutral, and pH 30 is violet, and more than the 30 which is violet, so therefore it will be given only from 0 to 14. So these are the pH steps which contains the standard color, and thereby the pH paper, sample paper, which can be tested with the standard papers to find out the exact pH of this. Then you can also use the litmus paper. So here the litmus paper is also useful to determine the pH of a solution when you do not have the pH meter available. So proper storage of these litmus paper is very much essential to maintain the quality control. So for example, a rough measurement of the pH can be obtained using these pH papers or the indicators, because pH changes colors with little varies. So these indicators have limitations on their accuracy, because pH will not be giving much exact accuracy, but you can get the broad range of the pH. So these kind of colorimetric methods which give only the broad way, but you cannot expect the accuracy, the exact pH. So these are the references. Thank you.